Hi, good afternoon, everyone. My name is Dan O'Connor. I'm the president and CEO of OncoSec Medical. I'd like to thank Dave Levine for having the company at the conference today. Really appreciate that and appreciate Dave's support over the years. Also want to thank you for attending the conference today. I'm going to go through several slides. I'm going to go through our corporate slide deck. I'm not going to hit every slide in the deck, but I am going to focus on a couple key slides that I want to bring to your attention. I think I would probably just start by saying right now is a pivotal moment for OncoSec because today and yesterday we released data on a program that we've been running for 3 years, and I'll describe that program in detail during the presentation. It's a very important clinical study, and it's exceeding our expectations in terms of what we hope to see from a patient response rate perspective. So I'm going to go ahead and start sharing my screen, and we'll get going. So OncoSec, again, is a cancer immunotherapy company. Sorry, it's scrolling a little quickly here. This slide is regarding our forward-looking statements. During this presentation, I may be making some forward-looking statements. So please take a moment to read the slide. I appreciate that. Sorry, this is really touchy here, so I got to get this under control. Let me try something else. It's going very quick. Okay. So this is our highlight slide. It's telling you the basic information about the company. We have a market cap that's actually significantly out of date from this slide, and we will update the slide. But today, if you looked at our market cap, you'll see it's about 145 million and a stock price of about $5.25. We've seen a very significant increase in the stock price over the last year, over the last 6 months, and over the last 3 months. And that's principally due to the data that I'm going to share with you during the presentation. We are a cancer immunotherapy company, which means that we're using the body's own immune response to fight back cancer. And we're doing that in combination with Merck's Keytruda. Merck's Keytruda is probably one of the most important drugs in our lifetime. Again, I'll describe that during the presentation. So our lead product candidate, you see this in the 4th bullet point here, is called Tavo. It's tavokinogene telsoplasmid, which is really too hard to say. So we call that Tavo for short. And that's what we're combining with Merck's Keytruda in late-stage metastatic melanoma patients who got Keytruda and didn't get a benefit. Again, I will describe that during the presentation. Sorry, this is just really touchy here, guys. So let's start with the premise. We have something that's happened in cancer immunotherapy since these technologies were first envisioned as the best way to potentially treat a cancer patient, specifically with their own natural immune response. So the whole premise behind cancer immunotherapy is taking the patient's own body and using that to eject the cancer instead of using chemotherapy, or radiation, or surgery, and all those tools that are effective but have major consequences associated with them. I don't think there's anybody that's listening right now that doesn't know somebody that has had to endure chemotherapy. In fact, may have been something that actually ended up causing them to die. I have a very good friend that was treated with chemotherapy, and it wasn't the cancer that eventually killed him, it was the chemo. So cancer immunotherapy, the idea is using the body's natural immune response and harnessing that to eradicate the cancer. The biggest and best programs that have evolved in this space, especially over the last 5 years, is a drug called Keytruda, and it's marketed by Merck. And Keytruda is what's called a checkpoint inhibitor. And the way that Keytruda basically works, and people have all sorts of metaphors to describe how it works. I actually like to describe it this way. And it's not too sciencey, so just hang with me for a second. When a person has cancer, there's a type of cell called a T cell that eradicates the cancer. They sometimes call them cytotoxic T cells or CD8 positive T cells, but they're T cells. And T cells are like mini grenades. They explode on a tumor when they encounter the tumor. And T cells, unfortunately, have off switches. And cancer turns the T cell switch off. Keytruda puts a cover over that off switch on the surface of the T cell to prevent it from being turned off. It's a monoclonal antibody that binds to that receptor that would otherwise be able to be available to turn the T cell off. So when Keytruda is used, the patient's T cells, those natural cells, part of our immune response, and actually can eradicate a cancer and kill a cancer, are kept on. And that's important because those cells can now traffic to the tumor to do their killing job. But as this slide is saying, there are a lot of patients that get Keytruda, the off switch is blocked, but when those T cells get to the tumor itself, the tumor itself is immunologically suppressed or cold. And that is not capable of actually being attacked by that T cell, even though the T cell switch is on. And so, on one hand, you have this incredibly successful drug called Keytruda. BMS has got another version of the same type of drug. It's called Opdivo. I'm sure you've seen commercials for these drugs on television. Keytruda generates over $1 billion of monthly revenue, and it's just getting going. That, I think, gives a kind of a gauge in terms of how successful this drug is and how important it is. And it only helps a minority of the patients that get it. Not that every patient is going to experience that T cell being kept on feature, but those patients who don't get a benefit are because their tumors are immunologically cold and not receptive to those T cells which have been kept on. In other words, those T cells traffic to the tumor, they get to the tumor itself is immunologically suppressed and turns those T cells back off or prevents them from getting in and killing. If one were to make a list of all the things that could potentially change a cold tumor into hot, our drug, IL-12, would be at the tippity top of that list. That's because IL-12, or interleukin 12, is a cytokine that our white blood cells naturally make that tell the immune system to inflame. Okay, they don't block they don't create T cells per se. They don't block that interface like Merck's Keytruda does, but instead they signal inflammation. It's called a potent pro-inflammatory signaling cytokine. And so when you're solving for the cold tumor problem so that Keytruda can work in not just the minority of the patients, but the majority of the patients, and you're looking to solve for that problem, IL-12 is at the top of the list of the things that can help. Now, OncoSec recognized that a decade ago. In fact, they recognized that IL-12 could be used on its own as a monotherapy. And that's where the drug was first used by itself in lots of different types of cancer. Over the time that the company's been in existence, Merck's Keytruda got approved where? Melanoma. It was the very first drug approval, skin cancer. And interestingly, it was a study, just like the study we are now conducting with Merck, a single-arm registration-enabled study under accelerated approval where Keytruda was first approved. And study was a 24-patient response rate, which I will come back to because I think that's relevant to our data that we're putting out now. But just to continue to the background information, let's recap. Keytruda is a very successful drug. It keeps T cells activated. T cells get to the tumor and they get turned off because the tumor is immunosuppressed. Our drug, IL-12, can change a cold tumor into a hot tumor so that when you use Keytruda with the patient, you can get a response. And by response, I mean their tumors will shrink. So we have a system that's basically been used in research, on bench research for a very long time, because it's an efficient way to get DNA inside of cells. So let's talk about how our technology specifically works. We use a system that people will call electroporation. Really, I like to think of it as an electronic gene transfer system. Because what we do is we take a strand of DNA, which has been coded, C-O-D-E-D, coded, so that it will make IL-12, okay? So we code onto that DNA the sequence for human IL-12. And that DNA itself can't do anything. DNA in and of itself is inert. So, that DNA cannot make IL-12 unless it's inside of a cell. So DNA on its own can't do anything. It's kind of like a car engine. If it's sitting on the curb, it can't do anything, but you put it inside of a car, it can move the car forward. Similarly, a plasmid of DNA with IL-12 on it outside of a cell can do nothing. It's inert. It'll get destroyed by the immune system. But getting it inside of the cell, and once it gets inside the cell, it can tell the cell to make IL-12. And that's what we do. And we use a system which is disruptive, clearly disruptive, because it's not putting a drug intravenously into the bloodstream, which is pretty much what everybody is used to doing when we talk about cancer treatments. It's putting the treatment into the tumor itself. It's putting that strand of DNA I just described, coated with IL-12, and you see as a step one on this slide, directly in a syringe into the tumor. So if I were a patient on our study, or a patient going to receive our drug, I would be a melanoma cancer patient. And by the way, we can treat many different types of cancers, but we're furthest along with melanoma. And I would go to my oncologist and he'd say, Dan, hey, I'm sorry. You've got late stage skin cancer and it's on your skin and it's inside your body. Okay. That's called metastatic disease. And he or she would say to me, I am going to treat you with a drug called Keytruda. Okay. I can't give you chemotherapy because it's spread throughout your body. I'm not going to necessarily use radiation and it's too big to surgically resect. It's all over the place. It's late stage disease. Unfortunately, we didn't catch it earlier, but you have it and this is what we have to deal with it. So we're going to use Keytruda. And or we could use Opdivo, but we're going to use Keytruda because that's what I use in my practice. I'm kind of envisioning what the doctor would say. And so the doctor would put me into an infusion clinic, put a bag with Keytruda, and I would get intravenous Keytruda. And in that conversation, I would probably say to the doctor, logically, hey, well, what are my chances of getting a response? Like, is this going to make these tumors go away? What are the chances of that? It comes back to that earlier slide, about 30%. 1/3 of the patients see their tumors treated with Keytruda. And so off I go, infusion clinic. I then come back, I get Keytruda, come back to the doctor's office and say, where are we? What's going on? The doctor will look at my skin and he'll take pictures of inside of my body and will say, and unfortunately will say to me, for our patients, hey, Dan, sorry, Keytruda didn't work. It actually didn't help you. And of course I'd probably say, well, why not? And he'd probably explain what I just explained to you, which is that the drug, kept those T cells alive and kicking, but my tumors were immunologically cold, in fact, too cold to get a benefit from Keytruda. So, at that point, the doctor might say, you know what, I have a clinical study that I'm going to put you on because I think we can solve for that cold tumor problem with a drug called Tava. And what would he or she do? They would enroll me in the study, obviously, but take that same tumor on my arm, inject directly the DNA plasmid for IL-12, and then use an applicator, looks like this, putting it on the skin, pushing it into the skin, and for 1 second, delivering a pulse of energy into that tumor. When that 1 second pulse of energy occurs, the DNA, like the car engine, moves outside the cell and into the cell, but it happens to millions of cells. And then all of those cells become like IL-12 factories for us. So they all start making IL-12. And IL-12 itself signals something called interferon gamma. Again, not too sciencey here, but IL-12 signals gamma, gamma signals 12. You get this robust production contextually in the tumor microenvironment, the tumor itself, of IL-12. Importantly, that then signals an immune response, not just in that tumor we treated, but throughout the body. And that's our clinical study. It's taking those patients who got Keytruda, who didn't get a benefit, who get our drug and potentially could get a benefit because our drug turns cold tumors into hot tumors. We've used our drug both as a single agent and now in combination with Merck's Keytruda. In fact, we're on our fourth clinical study with Merck and Keytruda. We first used it in what's called the 102 study. I won't go into these study details. That gave rise to the study that we're doing now, called KEYNOTE-695. That study you see at the top of this slide on slide 10, it is a very important study, and it's the study by which we just came out with data actually today at the SITC meeting, later on this afternoon, and yesterday in a press release that the company issued. So what is that study? And I'm going to walk through that data with you. First, the study has been designated by the US FDA as a fast-track program. So Tavo in checkpoint refractory, so patients who got Keytruda and it didn't work for them, metastatic inside the body, melanoma, skin cancer, Tavo has been designated a fast-track program in that patient population. So we're doing a study called KEYNOTE-695. Merck brands all of their important studies Keynote studies, key obviously being a play on words with Keytruda. So this is a 100-patient study that started literally 3 years ago. November of 2017, we dosed our first patient. And here we are today having almost full enrollment in the study and being able to provide the data that I'm going to show you in this waterfall plot. Sorry for the slides jumping around, guys. If I can get there. There we go. So, I don't know if you're familiar with these type of slides, but this is a really important way that data gets presented in oncology studies that are looking to shrink tumors. Everybody's familiar with a graph, and you have your y-axis. I like to remember that y goes to the sky, so that's the one that points up, and your x-axis that goes across. This slide is showing us patients whose tumors percent change from baseline, meaning that when they came onto their study, we measured their tumors. They were at 0. That was your baseline. Not that the tumors were at 0. There might have been tons of tumors all over the patient's body, but that's your starting point. That's your baseline. And then as the patients on the study, you see, going up the y-axis, you see tumors that are growing. So on the left side of the slide, you see patients who didn't get a benefit from our drug, right? On the right side of the slide, you see all those patients who saw their tumors shrink drastically. In fact, the response rate that we've reported in the first 54 patients evaluated is best in class. If you go out and compare this data set with any other data set that's been reported in checkpoint refractory metastatic melanoma, this is the best this is the best data that has been displayed in this patient population to date. The only other comparator that I think is relevant is a company called Iovance, and they have something called Cohort 4 for a TIL therapy. Iovance, by the way, with their program is about a $5 billion company. Again, this is why I think you're seeing our stock price start to really rise, because people are looking at Iovance and saying, gee, they had a 32% response rate, but there's some issues with their data, but 30 and 32% is roughly equal. They did not have the CR rate. You see CR rate here. That stands for complete response. These are the patients in green blue, that saw their tumors go completely away. You also see 2 of the patients here who saw a 100% reduction, but their doctors couldn't call it a complete response at that point. One patient, they had taken a piece of the skin to see if there was any cancer in the tissue around the skin, and they saw some questionable cells, so they didn't call it a complete response. Another patient had a very small, I think 3 millimeter enlargement of their lymph. Wasn't considered to be cancer, but they didn't want to rule it out, and by the rules of RECIST, they couldn't technically call that a complete response. But they did see their tumors go away 100%. So in the first 54 patients, we saw 5 patients who saw their tumors go completely away. And importantly, those responses are maintaining. Okay, so when I think about that, to me, it's very compelling, because I'm going to share a slide with you that's actually not in our slide deck. Give me 1 second. I'm going to see if I can locate it. Seeing this, but I did a quick, apples as apples to apples ish as you can get, and obviously these aren't head-to-head studies, so you can't make direct comparisons. But if you look at the Iovance study, which is this cohort 4 here, that's about as close as the type of patient we're evaluating as you can get. In fact, Iovance is going to be taking their data to FDA around the same time we are looking for a drug approval, and they had given an interim update of 68 patients out of 75 that they're going to report on. They in fact dosed 89 patients to get to 75 evaluable patients. They have a very, very high dropout rate in their studies, which is about 20%, and they don't report on those dropouts. And I actually think that's a potential issue, because it makes their data look higher. They make the denominator look smaller. But be that as it may, that's how they report it. And, we came forward with 54 patients, as I just described, out of 100 patients to target enrollment. We're almost at full enrollment now. Enrollment has accelerated greatly over the last several months, which is fantastic for us. And, we need our data to mature. We need at least 6 months of evaluation before we can even, call a response on a patient. So we have to see the data mature. As we reported out on the first 54 patients, we saw this 30% response rate. Iovance was a 32% response rate. 22 out of 68 were 16 out of 54. Interestingly, well, not interestingly, they didn't report it by RECIST, which is the standard, because they were also reporting 2 responses that had not yet been, excuse me, confirmed. So, gotta take a drink. Sorry, 1 sec. So, bottom line, it's going. Too much talking. Bottom line, the response rates. Sorry. One more drink. think I got something caught in my throat there. At any rate, 2 unconfirmed responses. Again, kind of unconventional to report unconfirmed responses. If they didn't report those, the response rate would have gone below 30. At any rate, we reported all confirmed responses by the industry standard of RECIST. So I think we compare very, very well, especially on the data sets. Again, you can't do direct comparison because they're not head-to-head studies. The only area we don't compare well with them is market cap. But I think, importantly, I think that's now changing. And hopefully in the process of changing with this data. Another area we highlighted is safety. They don't actually report the treatment-related adverse events. They report a different way. They report a treatment emergent. Treatment-related are those adverse events that relate to your drug. So it's akin to a treatment emergent, but I think the better way to think about it is treatment-related. But they didn't provide that information with respect to this cohort 4. Ours was.
Hey, Dan, you got about 5 minutes, okay?
Okay, great.
Okay.
Thank you, Dave. Thank you, sir. 5.4% grade 3. They have what's called almost 100% grade 3 and 4, which is, everybody's going to be in the hospital. In fact, with their technology, you have to go into the ICU to get prepared to take it. It's takes 6 weeks. There's a lot of issues with it, but be that as it may, I'm not trying to criticize them at all. I think, they have their data and they have a program. I think it's interesting about it is it's a good comparator for us. I like it as a comparator because if you assume that they're fairly valued, you have to assume that we're undervalued today. So let me just toggle back to the slide deck itself. And, hopefully you can see this slide. Can you tell me if you see that? That's great, Dave, right? You're seeing that? Good. Let me just take you to another couple key slides. This slide is showing what we call our visceral lesion applicator. So we have taken that applicator I showed you before, this one, which has these micro needles that look like that, right? Those small needles. And we've made it even smaller so that it can fit inside a syringe. It's basically, you can see it in this slide. I don't know if you can see it there, but it expands to 4.5 millimeters, but it's inside a 2 millimeter diameter, 1.99 distal diameter. So this is the needle that we put the DNA through. And here's where we create the electrical field I described earlier. But we're doing this inside of liver and lung lesions. That's something that nobody else can do. It's delivering a gene and basically a gene transfer system or gene therapy into a lung lesion or a liver lesion. And now we're looking to get this program started next year, into the clinic next year. And there has been a lot of interest on what we're doing in this program. Another program that was given an IND by FDA this year, and by the way, we're one of 8 companies, I think, when I read this in MarketWatch article recently, that has is doing or is positioned to do a human study in the United States against COVID-19. And we, many months ago, in fact, almost a year ago, started the process of thinking whether or not our TAVO or IL-12 could enhance a traditional vaccination approach. The idea got shared with NIH, and they almost immediately licensed to OncoSec their spike protein, the same one Moderna is using. And we're using it as a DNA platform. Moderna is using an RNA. And then I should say we shared the same concept with Providence cancer researchers, and they came back and said that they wanted to file an IND with the FDA, which is asking permission to use human patients or not their patients because they're healthy volunteers, but human healthy volunteers to try this approach. But our thesis here was not to try to compete with Pfizer or the big players in the infectious disease space or the coronavirus space, but really to see if the addition of TAVO could make those vaccines better. History has shown that the first vaccines that are used that come out to the masses, don't necessarily eradicate the disease. They tame the disease, but they don't build immunity for a patient so that if they get exposed a year later, they still have immunity against it, right? That's called memory. And, we've seen hints of data from Pfizer and others around how many neutralizing antibodies they're able to create, a 90% efficacy, but they're not sharing how long that lasts for. It's called memory. So we think the addition of IL-12 as a next generation vaccine after that first wave of vaccines could be a good thing. And at any rate, we're not spending a lot of money on it. We've really given it to Providence and the cancer researchers there who said, hey, would you let us run a clinical study with your TAVO and the NIH spike protein and our equipment? And we said, sure, we'd be happy to. So that's a quick summary of the company. And we have our cash on hand was enhanced at the end of August with 14 million. This is our last reported cash position at the end of July. We burn about 2.5 a month, no debt. There's the share count outstanding. We have a very good team that I'm very pleased to be working with. It's a team of people that have actually worked in the past together in large part. And so, I'm very grateful that we're here and together. Here are some of the anticipated milestones coming. Probably the most important is finishing enrollment in the study, which actually I think I need to add to the slide. Oh, here it is, complete enrollment. And then gather the data and file for drug approval, which is what we're planning to do in 2021 towards the end of the year if things go according to plan. And that's on this slide right here. We're hoping to be commercialized by 2022 and filing at the end of 2021. So right now, the task before us is to complete enrollment in the study, let the data mature, gather the data, bring it together in a regulatory filing, file it, have it reviewed by the FDA, and hopefully get clearance to start selling this drug. So with that, I will conclude. And Dave, thank you very much.
Dan, since you're bumping up against lunch, I'm going to go ahead and take. So you went over your time.
Sorry about that. Sorry about that. I always do. I apologize.
I know. I'm just giving you the business. So I do have one question though. I think we've got Rick. Do you have a question?
Yeah.
We have one question. We'll go ahead because they're still setting up lunch here. So we'll go have you answer one anyway. Rick, go ahead.
Well, it would make sense to me that, you're planning on getting this drug completed in the next year, that you would have a large pharmaceutical marketing.
Louder.
Yeah. So has Merck given you a target price that they're going to buy you out at? Or...
No, it's a great question. Not yet, but this is the first time these data were shared, right? This is, it took a while to actually gather the data here. I didn't go through all the slides, but you see in, the slide deck, we have done a lot of work to digest this data. And now we're able to share that for the first time with pharma partners, right? So I expect actually that there's going to be a fair amount of interest around this data going forward. Merck, I feel like Merck is very happy with the way we're that we have the ability to expand our relationship. Again, we're on our 4th clinical collaboration with them. And when they provide Keytruda to us, it costs them millions of dollars. And, we could go buy Keytruda in the market because it's available. It would cost us millions of dollars. And frankly, it's a lost opportunity cost for them. They've given us, probably thousands of doses of Keytruda at this point. So they definitely have skin in the game. Our drug does not cost anything close to what their drug costs. We're paying the third-party costs, but ultimately, I think they're very happy with how things are going. They're expanding the relationship. I think if they had competition, if somebody else came in and said, we really like this data set, we want to do something with OncoSec, you might see Merck move.
Well, Dan, I wish we could talk with you more because I think there's a lot more to talk about here. I just want to say that, you had you at the conference a couple times over the years. And, as you've done that, I know this has been kind of tough and challenging along the way, and 2 steps forward, 1 step back sort of thing. But I will say this, that there were times when I think when I talked about your company and the stock was doing what it was doing, which wasn't good, they sort of looked at me like I had 3 eyeballs, which people have done before. So I'm sort of used to that. But I think that there's something to be said for perseverance and hanging in there. You've certainly done that. I mean, I think if people are looking for managers that do that, you've been, pretty darn persistent with this. And I think the data is really, really outstanding. And, I guess I feel like the next 12, 18 months in the company are going to be telling. Yeah. So with that, Dan, I appreciate you coming again. And we look forward to more good news. So
thank you, Dave. Thanks for bringing us in. And thank you for that last question. Thanks everybody for listening to the program. We really appreciate it.
Great.
Okay.
Thanks, Dan.
Bye-bye.